Welcome back to Dead Reckoning, the Human Life Expectancy channel. In our last two videos, we focused on how reading the genome and epigenome with genetic and epigenetic tests can enhance HLE and revolutionize human reproduction. But reading the genome and epigenome is just the beginning. Rather than just read the genome to identify conditions, wouldn't it be better to edit the code to actually cure genetic diseases? In this video, we'll focus on our advancing ability to edit and write the code of life, DNA, using genetic engineering. Based on recent advances, we can now make a number of edits to the genome that have the potential to increase human life expectancy. While genetic engineering is currently available only as part of clinical trials in the US, it holds promise for treating a wide range of diseases such as cancer, heart disease, and diabetes. Ultimately, reprogramming our genes may prove to be the most fruitful approach for enhancing HLE, and significant progress is possible within the next 20 years. Genetic engineering vastly strengthens our ability to modify Homo sapiens and other species through volitional evolution. And of course, this newfound power must be balanced with new responsibilities that are based in ethics and manifest in effective regulations. Again, much more on applications of genetic engineering and ethical and regulatory issues in future videos. For now, let's focus on 3 steps that are necessary to use genetic engineering. Step 1 is to identify the specific order of base pairs that trigger genetic conditions. Step 2 is to find or create healthy versions of genes. And then step 3 is to deliver the healthy genes to an organism. So for step 1, we have to identify the specific order of base pairs that trigger specific genetic conditions. This is done using genome-wide association studies, called GWAS, that observe various conditions from organisms with or without specific genes. For example, here's the results of a GWAS study showing that single nucleotide pairs, or SNPs, are strongly associated with kidney stone disease. As we discussed in our last video, many diseases and attributes are discoverable today. We can now screen for over 10,000 genetic diseases that are determined by single gene disorders. And in addition, we can test for approximately 40,000 diseases that involve more than one gene, such as heart disease, many types of cancer, and type 2 diabetes. We can also scan for missing or extra copies of chromosomes, for example, Down syndrome. And we can also screen for sex and multiple physical traits such as eye, hair, and skin color, and even lean muscles. In the future, as genetic testing improves and the cost of genetic testing decreases, researchers expect that we'll be able to screen for much more. Most traits having a heritability of 50% or more could be predicted with relative accuracy once we have about 1 million people with their sequenced genomes and life records available, allowing for the GWAS studies. So let's move on to step 2. How can we find or create healthy versions of genes? Finding healthy genes is done by either genetic and epigenetic testings, as we've just discussed, or by creating new healthy versions using genetic engineering, which we're going to cover now. So how do we learn to edit the genome? As so often seems to happen in science, the most revolutionary tool for editing genes was discovered completely by accident. It turns out that our DNA naturally evolved to store mugshots of viruses that infected us in the past. Our bacteria actively search for these viruses and snip them out of DNA as if with tiny scissors, providing a helpful defense. So now, by changing these mugshots to match unhealthy genes we wish to replace, we can leverage bacteria's natural ability to destroy viral DNA this way to gain an unprecedented ability to re-engineer the genome. Since these mugshots appeared in our genomes as clustered, regularly interspaced palindromic repeats of genetic code of offending viruses, scientists chose the abbreviation CRISPR as the name of this revolutionary gene editing technology. The compelling story of how we learned to do this is well worth reading, and it's told well by one of the scientists that actually discovered CRISPR, Jennifer Doudna, in her book, A Crack in Creation. Before CRISPR, genetic engineering required substantial expertise and expense. After CRISPR, genetic engineering came to be easy and inexpensive. With CRISPR, scientists can easily target the genes of their interest and make desired edits themselves using standard techniques, all within mere days and without requiring outside assistance. The fundamentals can be mastered even by high school students, and there are companies that provide the tools and necessary reagents over the web. A crowd-funded venture now provides everything you need to make precision genome edits at home for about $130. And CRISPR is getting better all the time. A recent study showed that CRISPR could potentially relieve chronic pain and associated opioid addiction that's estimated to affect between 19 and 50% of the global population. Another recent study demonstrated that CRISPR gene editing can even be done in outer space. Scientists hope they can eventually repair the genetic damage from ionizing radiation, which could help NASA and other agencies develop technology that shields astronauts and makes deep exploration of space practical. So, how safe is CRISPR? Jennifer Doudna, one of the creators of CRISPR, said, quote, "It's almost certain that germline editing will eventually be safe enough to use in the clinic. Our entire lives are spent at risk of random genetic changes, and the threat from them is arguably far greater than any threat CRISPR would pose." The risks of CRISPR are small, especially when compared to the potential benefits to humanity, but they are non-zero. One potential problem with CRISPR is what's known as off-target effects. In short, these are places where the scissors cut the DNA in places other than those targeted by the therapy. Since DNA is extremely long, 3.7 billion base pairs, and has many repeats, this is a real possibility. However, the magnitude of these off-target effects is currently small compared to the natural DNA replication error rate and additional mutations from radiation and chemicals, and it's expected to get even smaller as the technology improves. Also, while CRISPR has not been shown to create cancer in people who've participated in clinical trials, nor in animal studies, one recent study shows that a mutational process called chromothripsis occurs in a small number of the cells that have been modified by CRISPR. However, it appears that a single-strand editing technology may minimize this possibility going forward. Another recent study showed that new links between CRISPR and cancer genes, including p53, could prevent the accumulation of mutated cells without compromising gene scissors' effectiveness. So while the broad use of CRISPR in a clinical setting is not yet here, there's no longer any doubt among molecular geneticists that the technology to perform genetic engineering safely and efficient will be developed. Importantly, the CRISPR system also contributes greatly to genetic research. It's now being used to record genetic changes in a cell over time, identify specific virus strains, test for infections, spatially reorganize the genome, and perform a host of other functions. CRISPR can do for genetic engineering what the Model T Ford did for automobiles. By streamlining the process and reducing cost, genetic engineering is now widely available. As a result, it's now being used to engineer plants, animals, and humans in many ways, as we'll explore in our next video. It's being used to treat genetic diseases and cancer, create cheap, reliable mobile tests, and even potentially cures for deadly infectious diseases such as COVID-19. And it's also being used to design livestock and crops that are hardier, more nutritious, and more economical. It could even allow us to choose how humans will evolve and potentially even cure aging. In addition, there are also multiple other gene editing technologies being developed, including base editing, prime editing, retrom library recombinering, and RNA interfaces, just to name a few. Another study showed that by exploring the evolutionary origins of an enzyme used in the CRISPR gene editing system, researchers have unearthed more than 1 million other potential editors lurking in our microbial genomes. This increases our confidence that safe, reliable gene editing is highly likely in the coming years. So far, we've been discussing edits to somatic cells, which are not inherited by offspring and die once the individual with the modified cells dies. However, genetic engineering can also be applied to eggs and sperm cells, also known as gametes or germline cells. When this is done, all of the future offspring resulting from the modified individual will carry that modified trait, permanently altering the gene pool of that individual's species. Because of their ability to drive genes through populations, scientists call such edits gene drives. Gene drives are a revolutionary technology that allows bioengineers to drive new genes with their associated phenotypes into populations with unprecedented speed. Gene drives use specially designed genes that have the unique ability to increase their frequency in the population to almost 100%. Once a gene is engineered into an organism's genome, its offspring will receive a copy of the gene drive from one parent and a normal copy of a gene from the other parent. Then, during early development, the drive uses CRISPR to replace the normal copy, causing virtually 100% of the offspring to possess the gene drive, rapidly spreading the gene drives through the population. For example, gene drives are now being used in the US and elsewhere to attack disease-spreading populations of mosquitoes, and resulting in a crash in their population and reduction in the disease. While this raises rightful concern, after all, we can't have individuals deciding to eradicate mosquitoes or any other species on their own. Regulations require strict protocols and safety measures for gene drives, and most gene drives are made to be self-extinguishing, thereby eliminating uncontrolled spread. More about this in our video on the regulation of genetic engineering. Of course, the therapeutic promise of gene drives is to eliminate genetic diseases. From a therapeutic perspective, gene drives have two big advantages over somatic and body cell editing. First, by editing a single progenitor germline cell, such as a sperm or an egg, the changes propagate throughout the body, eliminating the need to deliver the edits to the billions of somatic body cells. Second, every future generation will benefit from those edits. Because germline cells can forever change the gene pool of the species involved, there are significant ethical and regulatory issues to be addressed, and we'll cover those in future videos. For now, let's move on to step 3. How can we deliver healthy genes to ourselves and other organisms? Altering genes in blastocysts is much more simple than altering them in adults. Simply change the genome and it propagates throughout the cells of the adult. However, for anyone who's already contracted a disease, editing germline cells won't help. Editing their somatic, differentiated body cells is the only way to go. For adults, we have to implant healthy versions of the gene into large numbers of existing somatic cells and avoid triggering the immune system to attack the newly altered cells. Doing this mechanically would be extraordinarily difficult. Fortunately, as with CRISPR, we found that nature has already found a way to do this. In the late 1960s, we found that we could enlist the help of viruses to introduce genetic alterations to somatic cells. Viruses are small infectious agents that replicate only inside the living cells of organisms. They've evolved over billions of years to enter cells of any type and replace the cell's existing DNA with their own DNA, which then produces more copies of the virus. The first documented attempt at this was in the late 1960s when an American physician named Stanfield Rogers studied a wart-causing virus in rabbits called the Shope papillomavirus. He noticed that this virus transferred its genetic information into the host and began to study ways of co-opting the virus to become gene delivery vehicles, or vectors, as doctors call them. These vectors proved to be remarkably reliable. Researchers working with viral vectors quickly got to the point where they could transfer genes into the target cells with nearly 100% efficiency. In the 1970s and 1980s, scientists discovered ways to isolate targeted gene sequences and copy them into DNA, enabling them to insert beneficial genes into viruses and remove harmful genes so that the viruses would not harm infected cells. In 1974, Rudolf Janesch was first to create a mouse containing edited genetic material using this delivery approach. Using this approach, the first genetically engineered synthetic human insulin was produced in 1978. Viruses were used to deliver the genes of insulin-producing cells to E. coli bacteria to produce insulin. In 1982, Eli Lilly went on to sell the first commercially available biosynthetic human insulin under the brand name of Humulin. Now, nearly all insulin is produced from bacteria or yeast. Human growth hormone has also been produced in genetically modified bacteria since the 1980s. And now, the vast majority of biological drugs are produced in bacteria, yeast, or in cultures of cells from humans or other animals. Then, in 1985, Oliver Smithies found a way to deliver genetic code that did not involve viruses. He was able to replace the diseased cell's genes with a recombinant version they had constructed by using homologous recombination, the primary process of DNA replication. This approach leverages the cell's natural abilities without any trauma to the cell from viral infection and can be applied to somatic or body cells or germline cells. He shared the Nobel Prize in 2007 for this discovery. Today, gene delivery can be accomplished in vitro, in glass, or in vivo, in living cells. In vitro delivery is easier than in vivo delivery because the editing can be done under laboratory conditions. It's also necessary for editing gametes, such as sperm and egg cells. In vitro also has the advantage of being able to sequence and test the altered genome for accuracy in genetic defects prior to insertion into the living organism. In vivo delivery involves implanting genetically engineered cells into living people, which then exchange nucleotide sequences with similar identical molecules of double-stranded or single-stranded nucleic acids using Smithies' homologous recombination. This is harder to do than editing cells in laboratory conditions. It also requires that the altered cells be delivered to the right target area of the body. In vivo delivery of genetic engineering is now a reality for us. In November 2017, a man in California became the first person to have genes edited inside his body to treat the metabolic disease Hunter syndrome. Since then, in vivo gene therapies have been used for liver disease, congenital blindness, hemophilia B, cystic fibrosis, and many other diseases. Recently, researchers have demonstrated for the first time that a CRISPR therapy delivered into the bloodstream can travel to desired tissues to make edits. A CRISPR therapy was designed to target the gene that makes a destructive protein in the liver. And after just one injection into the blood, the treatment found its way into the liver and resulted in reductions of the destructive protein from 80 to 96%. This is a big improvement over having to remove affected cells from a patient, edit out the mutation in the lab, and then place the cells back into the body to replicate. Another in vivo delivery approach is to take pills or shots that impact the expression of our genes. More on this in future videos. While we don't yet have the tools to reliably edit every cell in the human body, an increasing number of in vivo and in vitro approaches for effectively delivering edited genomes exist and are being used today. So in sum, over the last few decades, our ability to accomplish the 3 steps of genetic engineering has advanced rapidly. New sequencing technologies have made it much easier to conduct genome-wide association studies and identify DNA base pairs to change. CRISPR and other new editing technologies made it vastly easier and less expensive to create healthy versions of DNA, and homologous recombinant technology has greatly facilitated the delivery of edited genes to cells in our bodies. Together, these enhancements mean that genetic engineering is destined for wide use in plants, animals, and humans. In our next video, we'll see that it's already being widely used in plants and animals and is starting to be used on humans, offering the potential to cure diseases, extend human life expectancy, and even guide human evolution. In the meantime, please hit the like button below, subscribe to the Human Life Expectancy channel to make sure you don't miss our next videos, and post comments for the topics you'd like to have us cover. Thanks for joining us. Till next time, as we say at Human Life Expectancy Inc., hi hua! Please, do what you can to help yourself and help us all to live longer.